So this fetal tissue's lack of blood samples, how much of that could be attributed to sample preparation, as alluded by Alistair? And could you explain a little bit more why
Fetal what?
in the fetal tissues you don't see much immune interactions as much as in the adults?
Immune cell interaction?
Yeah, immune cell interaction with, say, epithelial cells in the fetus. Is that how much of that could be due to sample preparation?
That's actually a good question. We thought about it. We feel like the reason why we don't see that for fetal, and we see that in adult, that's probably a good explanation that this is not due to contamination, because we will worry about could that be just contamination of immune-related genes to the parenchymal cells like epithelial cells. But if you look into that, I mean, in fetal stage, we still have lots of immune cells, but it's just that the receptor and ligand interaction doesn't happen there.
Mm-hmm. Okay.
So I think it's actually we're happy with this result.
Oh. Okay, great.
Okay.
Thanks.
So what was the kind of correlation between the two? Like, do you see any discordance, a cell positive for an antibody and not showing up in the transcriptome, or vice versa?
Yeah, look, you do, but that's partly to be expected. I think it's a combination of the biology where RNA and protein don't necessarily correlate, as well as the technology. I mean, even markers like CD4, everyone who's done single-cell RNA sequencing will know that it often drops out. And so using purely statistical measures of correlation, it looks pretty bad. At the kind of cell level, there's very good concordance. So if CiteSeq tells us that this is a particular cell type, if we take the transcriptome data, it makes sense. So I think in general they're harmonious. There are going to be differences, and that's part of what we're trying to figure out now.
And you showed in the cryopreserved thing, you got the comparable number of genes, but aren't those cells dead, or you just do the nuclei? Is it based on nuclei, the genes which you see in the nucleus, or the cytoplasm thing?
So all the data I showed you is viably cryopreserved cells. These cells are all alive, and in fact we do a dead cell removal step before we do any of our single cell captures. So these are all live, intact cells that we're comparing between the fresh and the viably cryopreserved condition. Is that what you're asking me?
No, when you store the chunks at -80,
Yes.
they won't stay alive. I don't think so.
They are alive, yeah. So we can culture them, we can xenograft them into mice. It's
So you put chunks with media?
with
or
DMSO serum. So if
Okay.
you dissect into small enough fragments,
Okay.
you get those cells stay alive. You do lose some cells probably
Yeah,
at
yeah.
the very center, but the majority of the cells remain intact compared to a suspension.
Okay, yeah.
Great talk. I may have missed this, but are you introducing UMIs in this last protocol that you were talking about, or just thinking about like different amplification artifacts that you might get by doing joint amplification? Whether there's any way to correct?
Currently, we haven't implemented UMIs, but this is something that we're thinking about.
Just a question about your long non-coding RNA data. So you're also getting full-length reads for long non-coding RNAs?
No, these are not full length.
Beautiful work. So I was wondering, I wasn't quite clear, how many transcripts can you look at the same time in the same cells? And if you were to correlate, say, the dynamic distribution of these transcripts over time and how they change, are they functionally correlated as well?
Yeah, that's a really good question. So we didn't use MERFISH in this study, and we only looked at one transcript at a time per cell and its cognate protein. So in the same cell, we would look at the RNA and its protein product. But obviously with techniques like MERFISH, you can scale this to hundreds of transcripts. The relationship between the transcripts is another question that we didn't look at, even though, so these transcripts are related in that they're implicated in sort of the migratory processes of the cell, like some of them. And so we could look at their, relative positions, but we didn't do that in this study.